This is Maggie Avars, and this is my final exam. So the first topic I'm going to talk about is differentiating the bacteria, archaea, and eukaryotic domains. So the highest level classification is the domain, and bacteria, archaea, and eukarya all fall under this. So on the earliest models, the first cells formed soon after the Earth formed, and from those ancient cells, bacteria and archaea formed. And then for over like 1 billion of years, these were the only forms on the planet. But later on, the eukaryotic cell formed, and this is what defined the presence of a true nucleus. And this is why the term eukaryote literally means true nucleus. And while bacteria and archaea, they lack one and sometimes are referred to as eukaryotes. So bacterial and archaeal cells are prokaryotic, which means they don't have organelles in their membrane, and they are much smaller than eukaryotic cells, like about 10 times smaller. And eukaryotic cells, these are more complex internally. They have organelles and can form multicellular organisms. And these differences are the result of evolution, which has been extremely small changes over time. And evolution is not a guess or opinion, but it's well documented and a testable process that explains how the two lives of bacteria and archaea have evolved and formed a third one, eukarya. So now I'm going to talk about the structural and genetic differences. So one way bacteria, archaea, and eukaryotes differ is in their cell structure, and especially the cell wall. So bacterial cell walls, these contain peptidoglycan, which is unique to bacteria and a major target for antibiotics. And archaea, they do not have peptidoglycan. Instead, they use pseudopeptidoglycan and other types of polymers, which helps explain why many antibiotics don't affect them. And then eukaryotic cell walls will vary. So plants use cellulose, fungi, and chitin, and animal cells lack cell walls altogether. So their cell membranes also differ. Bacteria and eukaryotes they have ester-linked fatty acids, while archaea use ester-linked isoprenoid lipids. And these are more chemically stable and help archaea survive like extreme environments. And only eukaryotes contain membrane-bound organelles like the nucleus, the mitochondria, and chloroplasts, and a bunch of others seen in the picture. And their organelles differ between animal cells and plant cells as well, so they're very diverse. But genetically, archaea are especially interesting because their transcription and translation machinery shares similarities with eukaryotes, even though they are prokaryotic. But bacteria are distinguished by their unique ribosomal RNA sequences, which is why they're separated into their own domain. So bacteria, they are described as ubiquitous, meaning they exist almost everywhere on the earth. So from like extremely cold conditions, and they even exist inside plants and animals. And archaea, these are often associated with extreme environments like high heat or low oxygen, high salinity, and some live in moderate conditions, but they are able to survive in those extreme ones. And despite being microscopic, these organisms play absolutely essential roles in sustaining life. So they do nutrient cycling and maintaining ecosystems. And all bacteria and archaea, they are microorganisms and are always single-celled, but eukaryotes, they can be either single-celled or multicellular. So humans, plants, and animals are all multicellular eukaryotes, while some are fungi and protists. These are microscopic eukaryotes. And an analysis of ribosomal RNA shows that all organisms in the three domains share a common ancestor, but modern science has recognized that evolution is not best represented as a simple tree. So due to horizontal gene transfer, especially among the microbes, life is better described as a web where genes move between species, and this ongoing exchange means that classification systems will continue to evolve as new information is discovered. So now I'm going to talk about the various and differential staining techniques, because I loved learning about these, but I couldn't remember all the different details. So staining is done to make microorganisms more viable under a light microscope. So with positive staining, the stain actually binds to the cell structures, but with negative staining, it colors the background instead. And positive staining uses cationic dyes that have a positive charge and are attracted to negatively charged bacterial cell surface. So when under the microscope, the cells appear to be colored and dark against a lighter background. But with negative staining, everything is basically the opposite. So the cell remains unstained and appears bright and visible, while the background will appear dark. And this uses anionic dyes, and they have a negative charge and are repelled by the negatively charged surface of the bacteria. So this staining helps show the true size of the cell, because the positive staining may require some form of heat, which can distort the cells. So negative staining is better for showing the exact size. So now I'm going to talk about simple and differential staining. And my coloring is a little crazy, but I wanted you to see which were which. So gram staining can be positive, which shows up purple, or gram negative, which shows up pink or red. And this type of stain is the basis for many biological traits like bacterial taxonomy, cell wall structure, and identification of an infection. So acid-fast staining is used to separate acid-fast bacteria, which will show up as pink or red, and the non-acid-fast bacteria will show up blue. So capsule staining also is another type, and it helps visualize small microbial capsules that don't absorb most dyes. So for this, a combination of negative stain and positive stain is used and leaves the capsules a clear halo. And then there's also flagella staining. This reveals the tiny filaments that are used by the bacteria to move. So we talked about gram staining a little bit. Gram positive will show up purple, and gram negative will show up pink or red. And this type of stain is best for biological traits like the taxonomy, cell wall, and infection. And acid-fast staining is used to separate bacteria, which will show up as pink or red. For acid-fast or non-acid-fast bacteria will show up blue. And I just wanted to go into detail a little more about these, even though they were in the previous slide. So now I'm going to go a little bit more into capsule staining and flagella staining. So capsule staining will help visualize the small microbial capsules, and they don't absorb most dyes. So this is why the combination of the negative and positive stain is used, and you can see the halo around the capsule. And then we have flagella staining. So this is good to see the tiny filaments or flagella used for motility, and this is good for identifying the bacteria type. So based on the amount of flagella or the different arrangements, they are able to better identify what the bacteria is. So now I'm going to talk about how microbes receive resistance genes through horizontal gene transfer. And this is a word I've seen a lot throughout the course of the microbiology, but could never really remember what it is, so I thought this would be a good refresher. So horizontal gene transfer is basically the genetic recombination when DNA is shared between organisms rather than inherited from a parent. So this results in new strains and new genetic combinations. And this process contrasts with vertical gene transfer. This is where genes move only from parent to offspring, and it explains why organisms that acquire foreign genes are considered recombinants. So a key reason horizontal gene transfer is so effective in bacteria is the presence of plasmids, which are the small circular DNA molecules that replicate independently, and they often carry beneficial traits like antibiotic resistance. So in addition to plasmids, chromosomal fragments released from damaged or dead cells can also be transferred, and these fragments will be integrated into the chromosome to persist. And the overall result of horizontal gene transfer is rapid evolutionary change. So this allows bacteria to grow quicker and gain new abilities like metabolism is enhanced, enhanced virulence, and it also makes them more resistant to drugs. So this makes horizontal gene transfer a major driver of microbial diversity and its ability to adapt. So now I'm going to talk about conjugation. So conjugation is a form of horizontal gene transfer, and it requires direct physical contact between the bacterial cells, and this allows DNA to move through a temporary connection. So this process is driven by the F factor, and this is a fertility plasmid that encodes the machinery needed to form conjugative pilus. And pilus is what sticks together and allows them to transfer. So it brings the donor and the recipient cells together. And cells carrying F plasmids are designated F positive, but those that lack it are designated F negative. And during conjugation, the F positive cells transfers a replicated copy of the plasmid to the recipient. But in some cases, the F factor becomes integrated into the bacterial chromosome, producing HFR cells, and these can transfer chromosomal genes at high frequencies. And conjugation is medically significant because it enables the rapid spread of R plasmids, and these often carry resistance to multiple antibiotics or heavy metals and virulence factors, and these will dramatically increase the pathogenic potential of bacterial populations. So now I'm going to talk about transformation. Transformation will occur when the bacterium takes up naked DNA from its surroundings. And this makes it unique among horizontal gene transfer mechanisms because it does not require direct contact, whereas conjugation does. So only competent cells can undergo transformation because they have specialized proteins that can bind and transport DNA across the cell envelope. And once they're inside the cytoplasm, portions of the incoming DNA may be integrated into the chromosome, and this will allow new genes to be replicated and then inherited. So I'm going to talk about the Griffith experiment, and this demonstrated the power of transformation by showing that a non-virulent bacteria could acquire capsule genes from dead cells and then become pathogenic. So beyond its natural role, transformation is foundational in biotechnology, and basically foreign genes are intentionally introduced into plasmids and then taken up by bacteria to produce a recombinant proteins or to help study function of genes and cloning. So in general, transduction is random fragments of the host DNA are accidentally packaged into phage particles. So specialized transductions are more selective, and they occur when a phage excises itself from the chromosome along with adjacent bacterial genes, and these often include toxin-producing genes that can help increase virulence. So another major contributor to horizontal gene transfer is transposable elements, or jumping genes. And these can move within and between chromosomes and plasmids when they use enzymes. And these elements will usually carry antibiotic resistant or toxin genes, and they can dramatically reshape genomes. So when large clusters of virulence are transferred together, they form pathogenicity islands, and these allow bacteria to rapidly acquire disease-causing traits and are essentially leapfrog traditional evolutional pathways. So for topic 4, I have chosen to differentiate between catabolism and anabolism because when I saw this topic, I thought, I don't think I could recall that and go back and differentiate it. So I thought this would be a good refresher. So catabolism and anabolism are types of metabolism. So metabolism is basically all the chemical reactions and physical workings within the cell. And all of metabolism includes thousands of reactions. Most will fall into one of the two major categories, which are anabolism and catabolism. So, a short overview of anabolism. This is a metabolic process that results in the synthesis of cell molecules and structures. It is a building, bond-making process which smaller molecules are assembled into much larger macromolecules that are needed by the cell. And because forming chemical bonds requires energy, anabolic reactions usually require an input of energy, most commonly found in the form of ATP. And through anabolism, the cell produces essential components that are necessary for growth, structure, and function. Catabolism is the opposite of anabolism, basically. Catabolic reactions break the bonds of larger molecules and convert them into smaller molecules. So, these reactions often release energy as the chemical bonds are broken. And the energy released during catabolism is captured and used by the cell, so this helps support other cellular processes that come after it. And it's also important to know that there's links between anabolism and catabolism that allow the cell to operate efficiently. And energy and electrons released during catabolic reactions are transferred through internal carriers, and they're used to generate ATP, which then fuels the anabolic reactions. So, electron flow is also central to metabolism, and it allows cells to collect, store, and spend energy in the form of ATP or heat. So together, anabolism and catabolism ensure that cells can both build the necessary structures and obtain the energy they need to survive. So now I'm going to go into catabolism. So catabolism is the portion of metabolism that's responsible for breaking down organic molecules to obtain both energy and precursor molecules that are needed to help the cell function. So they use enzymes, and catabolic reactions, they often dismantle complex nutrients, which are obtained usually from the environment. And they break these down into simpler compounds that cells can use for energy production or as building blocks for anabolism. So during catabolism, cells generate ATP as well as reducing power in the form of NADH or FADH2, which are essential for transfer of energy within the cell. So catabolism usually begins with the uptake of nutrients, and these are usually commonly carbohydrates like glucose, and these are took up through cellular transport mechanisms. And these nutrients are then processed through a series of the catabolic pathway, and this is the most common of this is glycolysis. So glycolysis basically converts glucose into pyruvic acid while producing ATP and NADH. So pyruvic acid serves as the central metabolite, and that can enter different pathways depending on oxygen availability. So when oxygen is present, this is called aerobic respiration. So pyruvic acid is further broken down through the Krebs cycle and the electron transport system. And this ultimately transfers electrons to oxygen, and that is the final electron acceptor. This process yields large amounts of ATP through oxidative phosphorylation and fermentation. In contrast, fermentation and anaerobic respiration occur without oxygen. So this produces smaller amounts of ATP, which use organic molecules or non-oxygen compounds as the final electron acceptor. And catabolism also includes the breakdown of lipids and proteins, which are converted into acetyl-CoA or Krebs cycle intermediates, and these allow cells to extract energy from different sources. And finally, there's anabolism. So for anabolism, this is the formation of molecules, so macromolecules and cellular structures. So this involves building complex molecules from simpler building blocks like monosaccharides, amino acids, fatty acids, or nitrogen bases or vitamins. And these precursor molecules come from one of two possible sources. They may enter the cell from the external environment, or they may be synthesized internally through specific cellular pathways. And an organism's ability to synthesize these building blocks depends on its genetic makeup, and this varies among different organisms. So, an important feature of this are autotrophs. They can synthesize all cellular substances using carbon dioxide, minerals, and energy, but heterotrophs, like E. coli and yeast, these can build all the necessary cell components from minerals and a single organic compound source like glucose. So there's also streak parasites, which are very different from this. These possess limited synthetic abilities and rely heavily on host-derived molecules. So once precursor molecules enter the metabolic pool, they become available for polymer synthesis. And all anabolic reactions require specialized enzymes and ATP to form chemical bonds. So anabolism includes carbohydrate biosynthesis, which ensures adequate supply of glucose and other sugars for structural and storage purposes. But then there's also protein and nucleic acid synthesis. These are central to anabolic processes as proteins from enzymes, membranes, cell walls, and other appendages. So ultimately, anabolic activity supports cell growth and the duplication of cellular components and cell division with the energy that's supplied from catabolic reactions. So now I'm going to talk about the different isolation techniques, because this is one of the things that I remember doing in lab a lot, but don't really recall all the different techniques. So isolation refers to the third of the 5 I's and is basically about separating one bacterial species from all the others. So the main idea is that if a single bacterial cell is given enough space and nutrients, it will multiply into a colony. And for isolation to work properly, only a small number of cells can be introduced, and they must be spread across a large surface so the cells don't grow together. So to do this, they use a solid medium like agar, and this provides a firm surface to keep the colonies separated. So other materials used for this are like a petri disc and an inoculating tool, and these are essential to help transfer and distribute the cells. So when done correctly, isolation can allow individual microbes to be separated from a mixed population so they can be studied more accurately. So here's some of the different isolation techniques. So there are 3. There's the streak plate, the pour plate, and the spread plate. So in the streak plate, there's a small drop of culture used, and it's dragged across the surface of the agar using the inoculating loop, and it gradually thins out the sample, spreading it further apart within each section. And this basically allows the individual cells to grow into separate colonies. And then we have the pour plate technique. This works differently, and you dilute the sample in tubes of melted but cooled agar before pouring it into the petri dishes, which reduces the volume of the number of cells. So as a result, some colonies form deep within the agar, but some are spread across the surface. So now I'm going to talk about the different cultures. So once isolation has occurred and colonies have grown, the cultures must be evaluated. So a pure culture contains only a single known species and is most commonly used in studies in the lab because it allows precise control and observation of the microorganism. And then a mixed culture contains 2 or more known and distinguishable species growing together. So a contaminated culture occurs when unwanted and unidentified microbes enter a culture that was once pure or intentionally mixed. And then contamination commonly results from airborne microbes and improperly sterilizing the inoculating tools or accidental contact with surfaces or like skin or different tools. So it's important to be careful when isolating because contamination can occur. So now I'm going to talk about the role of microbes in the food and biotechnology industry. And I thought this would be a good one to touch on because we did the different discussions, but we never did questions or a PowerPoint. And I just thought this was actually kind of interesting. So in microbiology, they focus on how humans take advantage of the natural abilities of microorganisms to improve both daily life and the health of the environment. So one of the biggest areas where this shows up is in environmental and industrial applications, where microbes are used in waste remediation, sewage treatment, pollution control, and even mining metals. So beyond the environmental cleanup, microbes are used for a wide range of products, including foods, medical compounds, drugs, organic acids, vitamins, and even alcohols. And these applications work because microbes have evolved and highly efficient metabolic strategies that allow them to survive when nutrients are scarce or when they're competing with other organisms. So applied and industrial microbiologists, they have studied these survival mechanisms and then they will use them and manipulate them so that humans can use them. And a major way this happens is through fermentation systems. These involve a large scale and controlled culture of bacteria or yeast, molds, or algae, and they consistently produce the desired compounds. So these are used a lot. So now I'm going to talk about how these processes have kind of changed over time. So this biotechnology has been around for a long time and stretched back thousands of years ago, before we even knew microbes existed. So early humans discovered that grape juice can be turned into wine and that dough containing certain cultures would rise, unknowingly harnessing microbial fermentation. And these early observations were the foundation for modern biotechnology, and which now uses recombinant DNA technology to enhance the natural abilities of these microorganisms. So through genetic manipulation, microbes can be optimized to produce larger quantities of specific foods, drugs, vitamins, or just compounds. So today, biotechnology spans many fields. So we use them in industry, medicine, agriculture, food science, and even helping protect the environment. And most systems rely on carefully selected and altered bacteria, yeast, molds, and algae, and they're efficient in synthesizing many useful products. So now I'm going to talk about what I was really interested about was microbes in the food industry. So they play a crucial role and intentional role in the food production through fermentation, which is used to make everyday products like bread, cheese, beer, yogurt, and wine, and pickles even. So in many cases, specific starter cultures of bacteria, yeast, or molds are added to ensure that the results will be consistent and predictable and in a good quality. So these microbes contribute to the different flavors, smells, or textures and appearances of food by producing gases, organic acids, alcohols, and enzymes during metabolism. And many fermentation are synergistic, which means that multiple microbial species work together in stages to transform raw ingredients into finished products. So because large-scale food production depends on these processes, there's a lot of effort that goes into maintaining the pure cultures and eliminating contaminated microbes that could spoil the product. And across cultures, microbes are responsible for the distinctive characteristics of these traditional foods, turning them into what might seem like decay into many different flavors and smells. So there's also microbes in water treatment and to help protect the environment. So microbes are essential to treatment of water and protecting public health and the environment. So drinking water is purified through filtration and chemical disinfection, while sewage treatment also relies heavily on microbial activity across 3 different stages. So during secondary treatment, diverse communities of bacteria, algae, and protozoa aerobically degrade organic matter. So this microbial metabolism converts the complex waste into simpler compounds like carbon dioxide, water, nitrates, sulfates, and phosphates. And then the remaining solid material, called biosolids or sludge, is often reused as fertilizer because of its high nutrient content. And this makes it a more sustainable alternative rather than just throwing it away. And microbes are also increasingly used in wastewater surveillance, where sewage is tested for microbial RNA to track the presence of the pathogens, such as the SARS-CoV-2, which provides early public health warnings. So now I'm going to talk about the role of enzymes in metabolism, and this seems like something that should just be engraved in my mind, but this was very complex, and I felt like this would be a good refresher as well. So we talked about metabolism, catabolism, and anabolism a little bit before, but just to refresh, metabolism is the collection of chemical reactions that are made possible through action of enzymes, and where they catabolize every step. So in the catabolic processes, enzymes break down organic molecules like glucose into smaller precursor molecules that can be reused for biosynthesis and energy. And then the same enzymatic reactions also generate energy in the form of ATP, NADH, and FADH2, which are required in large quantities for anaerobic reactions. And without enzymes, these reactions would occur way too slow to sustain the life of what they're in. And enzymes also provide reaction specificity, meaning in each step, metabolism is controlled and directed by a particular enzyme, which allows cells to regulate the flow and energy production efficiently. So now I'm going to talk about the enzymes in catabolic pathways. So once nutrients enter the cell, enzymes immediately take over the process through catabolic pathways. So glycolysis is the key example, where glucose is converted into pyruvic acid through a series of 9 enzyme catalyzed reactions, and each one will require a specific enzyme. So these reactions occur in a precise sequence where the product of one enzyme becomes the substrate for the next, showing how enzymes organize metabolism into very efficient pathways. And then along the way, they form and facilitate energy such as ATP, and then they reduce NAD plus to NADH, and this captures energy for later use. So after glycolysis, enzymes will continue to drive metabolism through the Krebs cycle, which consists of 8 more enzyme catalyzed steps that will extract electrons and carbon from the acetyl-CoA, and these enzymes reduce NAD plus and FAD, forming NADH and FADH2. And these carry high energy electrons to the respiratory chain. So the respiratory chain itself is made up of several enzyme complexes as well that are embedded into the membranes and pass electrons in a very controlled sequence. And the enzymatic electron transfer pumps protons and creates the proton motive force, which powers ATP synthase. And this is a complex enzyme that physically converts ADP and phosphate into ATP. So, finally, specialized enzymes such as cytochrome oxidase catalase, which is the terminal reactions that transfer electrons to oxygen, will complete the energy extraction role of enzymes in metabolism. So now I'm going to talk about the different clinical considerations in prescribing antimicrobials, because this chapter occurred pretty early on, but I always found it very interesting because I'd kind of learned about it in previous classes I took, but I got to see it on a much more in-depth level. So the primary goal of antimicrobial therapy is very straightforward in theory, but it's very difficult in practice. So the goal is to eliminate the infectious agent without damaging the patient's own cells. And this depends heavily on selective toxicity, meaning the drug targets microbial structures or processes while sparing human tissue. And another key consideration is whether the drug can reach the site of infection, since some body compartments like the brain or joints or skin can limit drug penetration. And the drug must also remain active in the body long enough to clear the infection while still being broken down and excreted safely. So, infections involving biofilms, these add another layer of complexity because microbes can be embedded into biofilms, which often respond differently to antimicrobials than free living cells do, because no drug is able to perfectly meet all the criteria. So prescribing antimicrobials will involve a balance of effectiveness and safety, and also practicality to reach the best possible compromise. So before starting antimicrobial therapy, clinicians will ideally determine 3 things. So the identity of the infectious agent and the susceptibility to drugs, and then the overall condition of the patient. So identifying the infection as early as possible is critical, especially collecting specimens before antimicrobials are given, since treatment can eliminate the pathogen and then complicate the diagnosis. And in some cases, therapy must begin empirically based on symptoms. So gram stain results or epidemiological patterns, even though it is not ideal. And susceptibility testing, this helps guide the drug choice, so particularly for organisms that are known to develop resistance. So using methods like the Kirby-Bauer test or determining the minimum inhibitory concentration, or the MIC. So these tests will show which drugs inhibit growth in the lab, but they also must monitor the patient because in vitro effectiveness does not always match in vivo outcomes. And I forgot what this means, so that's why I included the picture, because this also helped me gain all my knowledge back. So treatment failure can result from poor drug penetration and resistant microbes not captured in testing, and also mixed infections or improper patient use of the medication. So, beyond microbial susceptibility, patient-related factors play a major role in antimicrobial prescribing. So, the therapeutic index is a way to help clinicians compare the drugs by showing how close the effective dose is to the toxic dose, and with a higher index offering a wider margin of safety. And then closely related to that is the therapeutic window, and this describes blood concentration range where the drug is effective without causing harm. So physicians must also consider individual patient factors, like do they have allergies, what's the state of their liver or kidney, do they have diseases, their age, and pregnancy, since these can influence drug metabolism, excretion, and their overall safety. So drug interactions are another concern because some combinations will increase toxicity while others act synergistically and allow lower doses. So even when selective toxicity is achieved, antimicrobials can cause adverse effects and include organ toxicity or allergic reactions. So basically, antibiotics can disrupt the normal microbiota and sometimes lead to superinfections. So this includes the C. difficile associated with colitis or yeast overgrowth, making a microbiome preservation an increasingly important clinical consideration. So now I'm going to talk about the role of Koch's postulate and identifying the etiology of certain infectious diseases. And I chose this because this is another word I feel like I've seen throughout the course of microbiology, but had forgot it after I learned it and wanted to remember what it was before moving on. So the central purpose of Koch's postulate is to identify the etiological agent, meaning the exact microorganism that is responsible for a specific disease. So in 1880, Robert Koch recognized that simply associating microbes with a disease was not enough to prove germ theory. So he developed this experimental framework to demonstrate the true cause. And this experiment was especially important because science requires more than just correlation. It requires evidence that one factor directly causes another. And Koch based his work on careful experimental observations, particularly his studies on anthrax transmission in cows, which allowed him to trace disease from one host to another in a very controlled way. And the postulates were intentionally strict so that they could withstand scientific scrutiny, and this ensures that the conclusions about the diseases and their causes were reliable. And then, because of this, Koch's postulate became a foundation of medical microbiology and transformed how scientists identify the causes of infectious diseases. So, Koch's postulate outlines a stepwise method for proving that a microbe causes a disease. So, first, the suspected organism must be found in every case of the disease, which establishes a consistent association. And then, after that, the microbe must be isolated from an infected individual, ensuring a pure culture, and ensures that only the suspected organism is being studied. So that culture microbe is then introduced into a susceptible healthy host where it must reproduce the same disease with the same signs and symptoms. And then finally, the organism must be re-isolated from the newly infected host shown to be identical from the original host. And in practice, this means carefully microscopic observation, laboratory identification, and confirmation that both the microbes and the disease outcomes match exactly every time. So there were some limitations on this, and although it seemed reliable for many diseases, they cannot always be fully applied because some pathogens like Mycobacterium leprae cannot easily be isolated or cultured, which makes it difficult to test in the lab. And the postulates also depend on having a suitable animal model that develops the same disease as humans, which is not always possible and also raises ethical dilemmas. And viral diseases present additional challenges because viruses often infect only a narrow range of hosts. And then another limitation that was seen is the growing recognition that many infections are polymicrobial, meaning multiple organisms contribute to the disease, which prevents fulfillment of these postulates. And then to address these challenges, scientists developed molecular Cox postulates, which focuses on identifying specific genes that contribute to virulence. But despite these limitations, the logic of Cox postulate, which demonstrates true causation rather than simple correlation, is considered the gold standard for determining whether a factor actually causes a disease. And then for my last topic, I am going to talk about the different physical and chemical means of microbial growth. And I really enjoyed this topic, but there were so many that fit into the different categories that I thought it would be good to go over them one final time. So, heat is one of the most reliable and widely used physical methods of microbial control, and moist heat is especially effective because it works by denaturing proteins, and which this permanently disrupts enzyme structure and cellular metabolism. And then methods like boiling, pasteurization, and autoclaving all rely on this principle, but they differ in intensity and their applications. So, dry heat is different because it kills microbes by oxidizing cellular components, which dampers membranes and proteins, but it generally requires a higher temperature and longer exposure times to be effective. And then autoclaving, this is the gold standard for sterilization because steam under pressure can penetrate materials efficiently and reliably destroy highly resistant endospores. And then we have pasteurization. So this is designed to reduce pathogenic microbes rather than completely sterilize it. So it improves safety while maintaining the quality of food. And then we have cold temperatures. So cold temperatures don't kill microbes, but instead they will slow their metabolism and reproduction down, which makes refrigeration and freezing useful for preservation rather than true microbial elimination. So now I'm going to talk about a little bit of the other physical control methods. So radiation is another important one, and this includes ionizing radiation through extensive DNA damage, through the production of free radicals that can break chemical bonds, and this makes it highly effective for sterilizing medical supplies and certain foods. But then we have non-ionizing radiation, and this works different because it uses ultraviolet light, and ultraviolet light works differently and creates thymine dimers in DNA, which can block replication and transcription. And although its effectiveness is limited by poor penetration. And then filtration physically removes microorganisms from liquids and gases, and it's especially valuable for materials that can't tolerate heat that's used in the other methods. So this is good for like vaccines and intravenous fluids. And then we have desiccation. This is one that I really didn't remember, and I was glad to relearn about. So desiccation inhibits microbial growth by removing water that's required for enzymatic reactions and cellular processes, which explains why dry food resists spoilage. And then lastly, there is osmotic pressure. So osmotic pressure is like using high salt or sugar. It limits the growth by drawing out water in high contents of salt or sugar environments, and this leads to plasmolysis and metabolic inhibition rather than outright killing the bacteria. So then we have chemical control methods. So chemical control of microbes relies on a big range of antimicrobial agents that exist as liquids, gases, or solids, and these include disinfectants, antiseptics, sterilants, and preservatives that are designed for different settings and different materials. So these chemicals are often prepared as aqueous solutions or tinctures, and they depend on whether water or alcohol is used as a solvent, which affects penetration and activity. So, selecting an effective germicide requires balancing factors like rapid action or broad spectrum activity, stability, and minimal toxicity. So, although no single chemical meets all those criteria. So, chemicals are also classified by levels of disinfection. So, there's high-level germicides that are capable of destroying endospores and serving as sterilants for critical items. But then there's also intermediate and low-level agents, and these are used for semi-critical and non-critical materials. And the classification of the item being treated ultimately determines which chemical is appropriate and ensures effectiveness while minimizing the harm caused. And then there's also chemical groups that can control microbes through distinct mechanisms. And the first I'm going to talk about is halogens. So these are widely used because they are strongly microbicidal and can be sporicidal with sufficient exposure and time. And then alcohols, they act by dissolving membrane lipids and denaturing proteins. So they are almost completely effective. They have about 70% concentration due to the presence of water needed for protein coagulation. And then we have oxidizing agents. So these include hydrogen peroxide and ozone. And these generate highly reactive free radicals that can damage proteins, membranes, and nucleic acids. So this makes them very effective against a broad range of organisms. And then we have detergents and soaps. So these primarily disrupt cell membranes and remove microbes mechanically, but these are mostly limited to low-level disinfection. And then lastly, we have aldehydes and gases. So these include like glutaraldehyde and ethylene oxide, and these are among the most powerful chemical agents because they alkylate proteins and DNA, which halts metabolism and replication, which then allows them to function as true sterilants under certain conditions. And then that is all for my PowerPoint. Thank you so much.